All right, on with the next few sections of chapter 17. So the next part is all about genomic libraries. Now, genomic libraries are really important, right? So this is how the Human Genome Project, right? This is basically, we made a library of the entire human genome, and we made it because we wanted to sequence it and we wanted to finally figure it out. Genomic libraries were really important like a few years back because we still hadn't really figured out how to sequence really large fragments, like really large sections of our DNA. That was still not something that we could do. So we had to fragment our genome and make it smaller and then actually be able to sequence it, right? So genomic libraries are basically, we would take our genome, bombard it with restriction enzymes, so we'd fragment it, and then each of these fragments were individually put into its own cloning vector, so that cloning vector could clone, right? We could get more of those DNA fragments, and then we would sequence them. This project, like at least the Human Genome Project, was pretty much scheduled to last about almost like 20, 25, I wouldn't even say 50, I don't remember, but a really long time because it's a very painstakingly long process. You have to like fragment the entire genome. Each single segment has to go into a cloning vector. You have to allow the cloning vector to actually replicate and clone your gene or the DNA fragment that you put in, and then you have to sequence it. And then after you sequence it, you have to put everything together. So basically add everything as if those pieces of a puzzle in a linear sequence and look at this entire human sequence, DNA sequence as a whole. But this was quite, this whole process was quite accelerated just by novel DNA technologies. Things got a lot less expensive, right? We could basically sequence thousands of base pairs. By the end of the Human Genome Project, we could sequence thousands of base pairs every day. And so it actually it took about, I believe it started in 1990 and it ended around 2003. So it was very, very quick. So anyway, so this is how we actually were able to sequence our DNA in the first place. Our human DNA was through genomic libraries. So it was constructed by cutting genomic DNA with restriction enzymes, just making a lot of different fragments, and then putting these fragments into their own individual vectors. Now, this vector that they used had to be one that could carry these larger fragments, right? So they actually ended up using bacterial artificial chromosomes because they could be bigger, they can hold bigger things, and they were a lot more stable than YACs or yeast artificial chromosomes. So that they couldn't use any normal like plasmid DNA vector. Whole genome shotgun cloning and new sequencing methods are readily replacing genomic DNA libraries. This is because now, since our sequencing methods have gotten so amazing and so much better, we don't really need to actually clone genomes or fragments of our DNA within DNA vectors in order to actually sequence them. Now we can sequence a humongous genome, right? Just from that, without having to clone it into anything. Before you had to make lots of clones of that small little fragment or that biggish, largest little fragment, lots of clones, and then you could sequence it and be actually sure of what you sequence. That's not the case anymore. Wow, we can just take the genome without having to do any crazy preparation. So this is, you can sequence the entire genome without needing to put it into a vector first. So they're a little bit of a past, not really. We still use them a lot just because they are handy, but again, sequencing is making things a lot better. There's also complementary cDNA libraries. This is pretty much similar to what genomic DNA libraries are, except that now you're not just putting the entire genome in a library. The genome would be your genes and all the rest of the non-coding DNA, right? That's actually the majority of your genome. All the rest of that non-coding DNA within all these vectors. Now you're looking at complementary DNA. So complementary DNA is basically the DNA that is made into a gene, the coding DNA. Right, so we would do this by taking our mRNA, okay, we take our mRNA and then reverse transcribe it with the help of the enzyme reverse transcriptase. After we reverse transcribe it, we'd have a single strand of DNA that hybridized to a single strand of that original mRNA. So with that, we then partially digest the RNA with RNase H. It's important to partially digest because in this way, you'd have little bits of RNA kind of still stuck to our single-stranded DNA. And these little bits of RNA would provide a free 3' end. Note how we have a 3' end here, a 3' end here, and another 3' end here. which, as you guys know, is critical for DNA polymerase to use. So DNA polymerase would then use those 3' ends to basically extend and like read this template DNA, this template single-strand DNA, and put the complementary DNA on it. So we'd extend it, put our complementary DNA. The RNA primers essentially would be removed at this point, and then through DNA ligase, we'd ligate all the small little fragments and actually covalently bond the nucleotides with the phosphodiester bond. So this was really important because we would take our mRNA, which was devoid of introns, right? Devoid of any kind of non-coding intervening sequence that we would normally find within our gene. This is mainly for eukaryotes, right? That we would normally find within our gene, and just get the DNA that was needed for genes. That's it. And then we would put all this complementary DNA within cloning vectors and just have a host, like essentially all of the DNA that is expressed, okay, all the coding DNA within this library. This is extremely really useful because not only do you know which genes what part of the genes is actually coding and not non-coding, but you can also figure it out and make complementary cDNA libraries based on a specific tissue, right? So you have a specific tissue and you make a cDNA library just on that tissue, and you see which genes are actually made and expressed within that tissue, or even within a specific time period. So it could be development, something like an organism during its still developing stage, right? As opposed to an organism which is at its adult stage. So lots of different things to work with there. Or even when you have a cancer, you could look at the different gene expressions and figure out all the cDNA, all the cDNAs, and therefore construct a cDNA library of the genes that are expressed and made into mRNA when you have a cancer. So it's really, really important, a really important thing. After we construct our library, a lot of times it's necessary to then recover a specific gene. So in all of the cloning vectors that we have a myriad of cloning vectors. Each and every single one of these cloning vectors has another type of fragment in it. Let's say you want fragment F, right? Fragment F out of a myriad of fragments. How are we going to actually recuperate this fragment F? And this is actually a really fun way to do this. So we use things known as probes. These are basically, a probe is a single-stranded DNA or RNA sequence that is complementary to your target, to your fragment that you want, so fragment F. So the way it works is as follows. I'm not going to draw. I was thinking of drawing it, but I'm basically going to draw this. So if these are all your colonies, remember each colony here, let's pretend that the bacteria in this point, each bacteria has the same kind of fragment because they have the same cloning vector inside of them, right? So this bacteria right here at the top, I'm going to make this a little bit bigger. This bacteria right here at the top, okay, has fragment A, right? This bacteria to the side has fragment B. This bacteria in the middle has fragment C. And this bacteria right here, this colony on all these bacteria in this one colony have fragment F because they have the cloning vector with fragment F. Fragment F, because I made it up right now, is a target sequence that you want. So what you would do is that you'd put a thin layer, you basically overlay this thin little, this membrane filter over your colonies. What that would do is it would just right above. That would transfer all of the colonies or some of the bacteria from here to your membrane. So now if you then flip over your membrane, right, and put it in a place like a medium where the colonies, the bacteria can grow, those bacteria are going to grow back to normal as if nothing happened. You just transferred a few bacteria from your original plate to the membrane. Once you do that, then you denature your DNA. So basically, when we say denature your DNA, that means you make it into a single-stranded format. Okay, so your DNA is no longer double-stranded. It's no longer bound to its complementary DNA strand. It is now single-stranded. So this makes way for a possible probe, a complementary probe, to go ahead and attach. So you have made an actual probe that has a complementary sequence to your fragment F. This probe can be RNA or it can be DNA, doesn't matter. So you put then this filter with your colonies that have denatured DNA inside a little baggie. Literally, it's an actual heat-sealed baggie, okay? Inside a little baggie, and in this little baggie, you dump in the probe. the probe is going to navigate throughout all of the DNA that it has here, right? All the DNA until it finds that one DNA that it's complementary to, okay? And we know that the probe, we have made it so that it's complementary to this fragment F that is found within this bacteria. And so after that, you can then do like X-ray film, autoradiography, fluorescence, or whatever you have you to allow for the probe to actually light up. After you rinse everything, right? Because you made the probe has hybridized, it's annealed. After you've made the probe put in and the probe has annealed, you then wash everything away. Any probe that hasn't attached, you want it to be washed off. Now at this point, the only probe that has attached is your probe to fragment F. And it tells you here that there's hybridization of the probe on this colony right here. This is indicated in this case by a spot on the X-ray film. You can use again autoradiography or fluorescence or whatever have you for different like probes. In this case, we're using autoradiography and X-ray filming. So your fragment F is here. And so you compare your little filter. You know that your fragment F is right at the bottom. You go back to your original colony, your original like library of colony, like library of, let's pretend this is a genomic library, and you're like, okay, this position of this colony is the same as this position in this colony in my original one. So I'm going to pick that colony with a little loop, and I'm going to grow it alone in another medium so I can analyze it, I can analyze that fragment alone. That's essentially how it works, right? Probes don't necessarily even have to be incredibly specific. If you obviously know the sequence of your fragment F, this is nice and easy. But a lot of times, like probes can even be like partially complementary. So we could use, if we wanted to see, for example, if, this is really random, I'm not sure why this is coming in my head, but if a cow, let's pretend we did a genomic library of a cow, and we wanted to see if this cow had one of the same enzymes that humans have. So we made the probe of the gene that encodes that particular enzyme, okay? I can't think of any enzyme right now, sorry. We made the probe of that particular human enzyme gene, encoding gene, and then we did this whole screening thing with that probe with the cow's genomic library. And so if our human probe, our human DNA probe, binds to with the gene that, not human as in the sense that it's only human, but it just has it derives from the gene of human origin. If it binds to another gene or colony here in the cow, that means that cow has a very similar gene to us. That means that cow has a very similar enzyme to us, and therefore probably undergoes a similar chemical reaction or metabolic process similar to us. It's a little bit weird, especially with the cow example, but hopefully you guys understand. If not, Zoom meeting, right? So libraries enable scientists to clone DNA and identify individual genes in the library, or identify genes that are like homologous between different organisms. Lots of different kinds of benefits to screening and finding your actual DNA. And then it's also a way of isolating your DNA so that you can then go ahead and work on it more, right? So the next section is the polymerase chain reaction. Dun, dun, okay, huge discovery by Carrie Mullis. So this was discovered in the late 1980s, and this revolutionized genetics, okay? Because prior to that, the only cloning methods we had were with those plasmid cloning vectors or those other cloning vectors that we looked at, a very tedious, time-consuming, and even sometimes relatively expensive means of cloning your DNA. And then after you clone your DNA, you'd have to target it, you'd have to isolate it, really complicated. The polymerase chain reaction skipped all of that. It's very cheap, quick, can amplify and clone your DNA in a matter of hours, very, very fast. So the polymerase chain reaction, or PCR, can produce many copies of a specific target segment of DNA. It's a rapid method of DNA cloning that eliminates the need to use host cells for cloning, so it eliminates the need of using cloning vectors. It pretty much finds your one segment of DNA that you want to clone in the sea or ocean, actually more, ocean of the rest of the DNA, and specifically targets and clones just that little piece of DNA, okay? So it does this through a 3-step process: heating, cooling, and replication. We'll look at how these steps work in just a little bit. This brings about a chain reaction that produces an exponentially growing population of identical DNA molecules. So it does require a few things. It requires, it requires DNA polymerase and primers, magnesium, which is just there to stabilize the enzyme, and nucleotides. This is because PCR pretty much works as if normal DNA replication. This is what it's mimicking. It's mimicking DNA replication. So it's using essentially everything, all the enzymes, okay, or the majority of the enzymes and the materials that you would need in normal DNA replication. That's basically what it is. Yeah, so let's look at what our PCR looks like. So there are 3 steps. The first step is 3 heat-controlled steps, right? The first step is denaturation. So at this point, we are trying to denature our DNA. So denaturing means, again, just making our DNA go from double-stranded to single-stranded. So we separate our 2 strands of DNA from each other. Remember, this target sequence is in the midst of all this ocean of genomic DNA, okay? And we only want this small target sequence. This is the only part of the genomic DNA that we actually want to amplify and clone. At this point, we have the second step, which is annealing. So denaturation happens around about 90 degrees Celsius, 95 degrees Celsius. Okay, that's because we need to heat up the DNA to an extraordinary amount of heat in order to actually denature it. Annealing then brings down the heat all the way down to about 40 degrees Celsius, 45 degrees Celsius, depending on the primer. Annealing is basically when we put down the temperature that low, primers now, right? Remember how in DNA replication, our DNA polymerase requires the use of a primer because we have a free 3' OH end. Primers now are able to anneal to the sequence that is flanking your target sequence. So yes, with PCR, one of the downfalls of PCR is that one of its downfalls is that essentially we need to know a little bit of the sequence of our target gene, because if we know the sequence, we can make the primer that is then complementary to the sequence targeting, the sequence flanking our target gene. So our our primers are flanking our DNA, and they're going to be extended and elongated with the use of DNA polymerase. And so you can see here that we're extending right where we need our target sequence to be extended to actually be replicated, right? So that extending phase is called extension. At this point, we increase the temperature a little bit to about 60 degrees Celsius, because this is where our DNA polymerase really works well, or the specific DNA polymerases in the PCR reaction work best at. It's their optimal temperature. And so at this point, with the temperature slightly more higher up, around 60 degrees Celsius, we can start elongating our DNA and actually synthesizing the complementary strand, synthesizing the complementary strand to that template strand. And so now at the end of this, we have 2 DNA molecules, right? And so 2 DNA molecules. Oops. So after we finish this one cycle, these cycles are repeated over and over and over again. So you see here, cycle 1 yields 2 molecules. Cycle 2 yields 4 molecules. I can't read this anymore because there's something in the way. Cycle 3, 2 of the 8 molecules match the target sequence and the right length. So here we're starting to get the actual amplicon that we need. The amplicon is just a part of the part of the DNA that the only target DNA that we want actually clone, and none of the rest of the stuff. Because you can see in, I'm going to change my video. Okay. So if this is the entire genome, right? I'm going to draw the second strand right here. And one primer adheres, and this is my target DNA. This part right here is my target DNA. So this is my target DNA on the opposite strand. One primer adheres to the right on one strand, and one primer adheres to the left on the other strand. That first cycle, after we do extension, this primer is going to go straight all the way. And not only is it going to replicate your target gene, but it's going to replicate the rest of the DNA as well. So at the end of this one section right here, of this one replication, we have the primer with this, and we have your target gene here with the rest of the DNA. But you see how this other part is not included, right? Same thing goes here. Our DNA gets replicated in this direction, and this rest of the genome is not included. only on the next cycle when this separates. So now I still have my target gene here, right? This happened, this basically, it looks like, hold on. I'm drawing this over here. So this is what it looks like at the end of that first cycle, okay? So now denaturation happens again. We have the splitting up of the two strands. Annealing happens again, and extension happens again. So these two strands separate. I'm drawing them like so, okay? These two strands separate. One primer is going to go here, flanking it on this side, and then the other primer, remember, we're flanking always a target DNA, so the other primer goes here. So when this primer extends in this direction, this primer is going to extend in that direction and then stop because there's nothing else to actually replicate. And so at the end, with DNA replication here, we only have this target DNA. See how this is the complement of this target DNA. This target DNA has been completely amplified. This is our single target DNA amplicon. And so the rest of the jargon of the DNA that we have is no longer found. And this happens in the third cycle of our PCR. So heat denatures and separates the strands of the double-stranded DNA. Cooling allows for the annealing, or basically the hybridization of short primers that are complementary to the sequence on opposite strands at each end of the target. And then basically then our DNA polymerase is going to extend the primers to elongate and synthesize the complementary DNA strand. With each successive cycle, the number of molecules doubles. So it's basically, essentially almost exponential. By about 20 cycles or 30 cycles, you get about 1 billion actual DNA clones of only your target DNA without the rest of the DNA coming into place. None of the other part of the genomic DNA has been replicated. It's just your DNA. This is all carried out, so you've noticed that there's a lot of highs and lows in terms of temperature. It's all carried out in a specific kind of apparatus known as a thermocycler. It's just something that can really, that you put your little tube of, your experiment in. So it's your PCR, your template DNA with your target genome, or your target DNA, obviously, and all the PCR concoctions like the primers and nucleotides, the DNA polymerase, everything in the magnesium, everything in there. And then it makes these it repeats these cycles. It repeats cycles of really, really high temperature, lower temperature, and then medium temperature. Higher temperature, lower temperature, medium temperature. Repeats them, pretty much. Depends on how many times you want it to repeat. But usually cycles are repeated about 30 times. Each cycle is between 2 to 5 minutes, depending on how long you want it to go. And so you can finish a PCR of about 30 cycles or 40 cycles in about 2 hours. So really quick, a really fast time, and in about 2 hours time, you can have the complete cloning of your target gene. Another thing you have to take into consideration with PCR is that we are also using, because of these crazy temperature fluctuations, we must use a DNA polymerase that is actually stable with these fluctuations. So the DNA polymerases that we usually use are DNA polymerases that we find in usually bacteria or archaea, or basically organisms that live in really hot environments. For example, Taq polymerase is one of the most common heat-stable DNA polymerases that we use in PCR. Taq polymerase is isolated from an organism, from a bacteria known as Thermus aquaticus. This bacteria lives in the hot springs of Yellowstone Park, so in really, really hot areas. Another polymerase that we use, because Taq polymerase sometimes has really bad proofreading ability. So another polymerase that we use, is PFU, which basically comes from Pyrococcus furiosa. Pyrococcus literally means a fireball, right? So crazy amount of heat it can withstand. Pyrococcus furiosa is an archaea, I believe, that is found in like a volcano in Italy, that lives within the depths of a volcano in Italy. So really, really high temperatures. All of these DNA polymerases that we specifically choose are very good, are very heat stable. They're okay with temperature fluctuations, and they're especially okay when temperatures rise up to 95 degrees Celsius, which is usually the temperature that we need in order for the first stage of the cycle, denaturation to happen, right? And then, of course, the other thing that we need are primers that are specific for the sequence to be amplified. So a little bit of your sequence needs to be known. You have to make it so that your primers are flanking your target sequence, opposite ends of your target sequence, a segment molecule, and one at the 3' end of each strand. So that's it. You could also, this kind of here shows you a little bit more about, I can't see. Yeah, a little bit more of how the PCR works. I will be putting a video in there because I feel like animation with PCR works a lot better. You need to know what a cycle consists of, what the stages of a cycle are, and you need to realize, you know, when they're repeated, what that means, and how at the end you only have one single target DNA sequence repeated a million billion times. So it says here 25 cycles can give you DNA copies of that same target sequence of more than a million sequences, essentially, more than a million times, more than a million copies. So I will link a PCR video. I'll link a PCR video to kind of show you an animation of it, because it makes a lot more sense when things are moving. I couldn't make things move in my picture. Okay, so that being said, there are limitations to PCR, even though as amazing as it is, and as much as like inexpensive and as great as it has become, there are some limitations. So first off, the advantages: it's very rapid, very sensitive. It amplifies specific DNA sequences from sample DNA, from small DNA samples. So we can even use it to find literally minuscule. It's very sensitive, so it can amplify minuscule amounts of DNA. So even DNA that we find in like old bones, like so molecular paleontology, like figuring out ancient DNA, right? We can actually PCR that DNA, that even though it's very, very minimal, or even like at a crime scene, very minimal amount of DNA is found, and it can still be amplified because it's very sensitive. However, even if because it's so sensitive, and it can amplify really minuscule amounts of DNAs, it's also very privy to amplifying contamination. So when you do carry out a PCR, you have to make sure that you're not accidentally contaminating it. Like even something like skin cells from a researcher, which hold DNA, right, could basically give you negative results or really bad results, right? It's minor contamination from other sources can cause problems. Another limitation is that you need to know a little bit of your information of your nucleotide sequence so that you can actually make your primers. And then something else is that PCR cannot amplify extremely long segments. It usually amplifies things about 1,000 base pairs long or a little bit more, but not to an extreme amount. Like you cannot amplify the entire human genome just with PCR. That's because the DNA polymerase has a very low processivity. If you guys remember what that word means, it just means that it doesn't stick onto its DNA, its template DNA, for a very long time. So because of that, it doesn't replicate things for a long time. It doesn't go on replicating the same strand for, you know, millions and millions of base pairs. It doesn't, because of its low processivity. It falls off, and so you don't have really long segments. So it's very specific and speedy, but it can't substitute gene cloning in cells to make large amounts of a gene, because some of the DNA fragments that we need to clone are really, really big. So it doesn't really substitute the entire genomic library aspect, for example. So PCR amplification occasionally incorporates errors into the amplified strands, and that limits the number of good copies and the length of the DNA that can be copied. But like that's kind of helped a little bit more with the different polymerases that we use. TAC is a little bit more prone to incorporating errors compared to PFU, but that's just something else. Still though, the processivity of both DNA polymerases is quite low, and so you wouldn't be able to really copy a really long segment of the thing, of the actual target. So instead, PCR is used to provide the specific DNA fragment for cloning. In this case, PCR primers can be designed to include restriction sites that allow the product to be cloned into a plasmid vector, and then the resulting clones are sequenced and error-free inserts are selected. So we can kind of tweak our the target sequence that we want to include something around the sequence that it normally wouldn't have. What does that mean? I'm going to stop the share because it's confusing me, and I'm going to change video. Okay, so I got a bunch more colors because I was seriously lacking in colors. So we know that in order to put a piece of DNA target inside a cloning vector, that DNA target needs to be cut with restriction enzymes, right? Restriction enzymes so that our target DNA can be put inside our cloning vector that we cut with the same restriction enzyme. But what if this is our DNA target, target A, and we don't have any restriction enzymes here? In fact, the restriction enzymes are really, really far apart, but we don't want the rest of this non-coding DNA, let's pretend, right? The rest of this DNA that we don't want to analyze. How are we going to put only this part of the DNA, this segment A, into our cloning vector right here in the multi-cloning site? Okay, how are we going to do that? We can do that through PCR, because with PCR, not only can we amplify this over and over and over again, but we can tweak it as well. We can tweak the ends so that it includes the restriction enzyme site. How do we do that? We do that thanks to the fact that primers, when they anneal to the DNA, the only major part that anneals is the 3' end. So the 3' end, I'm going to draw the primers in green. Okay, so this is my DNA with my target DNA right here. This is what I want to do. Okay, the 3' end of the primers has to be the actual complementary to the actual sequence that we want to do. But then the rest of the primer, the 5' end, does not necessarily have to be complementary. This is the only part, the 3' end is the only part that really needs to bind properly to our DNA target. So even here, we can make a primer that has its 3' end, which is I know it doesn't look like a case here, but let's pretend like about 20 base pairs that are complementary to our target DNA. And then the rest of the bases in the rest of the primer, so towards the 5' end, are not actually complementary to our DNA. What happens here is that upon replication, so replication, this whole part gets replicated, and then here, this whole part gets replicated. So now, I'm just going to continue this section right here, right? So I have this primer, and right here, my target DNA sequence with this rest of the DNA, okay? On the next replication, so this basically denatures. I have my primer here still, and my DNA target right here. Okay, in the next replication, my new primer comes here with that 5' end that does not replicate, that does not bind because it's not complementary. And when it extends, can you see that when we have extension and we have DNA polymerase, like DNA synthesis, it extends. Not only does it does it synthesize my actual DNA, the target DNA here, but it also synthesizes this DNA on the 5' end of the primer. And so if we continue this replication, what we're going to get is we're going to get a target DNA, right? And at both ends of the target DNA, we have this extra 5' end that is not part of the actual target DNA. And we can make this 5' end a restriction site. So this holds a restriction site. So now we have made our DNA target with restriction sites. This is a restriction site here and another restriction site here. Okay, so then since they have a restriction site, we can safely put it into we can cut it, we can cut the vector with the specific enzyme that constitutes the same restriction sites of our target DNA. Okay, and then we can easily put our target DNA in, even if our target DNA originally did not have the restriction sites flanking it. This is known as seamless cloning. I don't know if, seamless. Okay, so what are some applications of the PCR technique, of the polymerase chain reaction? So this is an extremely useful tool. It's the most widely used technique in genetics and molecular biology, and this is not an exaggeration. This allows for screening of mutations, including genetic disorders or infectious disease. Basically, if we know that there is, we know of a particular genetic disorder and we know of its genetic, the actual mutation, we can isolate the genomic DNA from a person and then make sure that we target that genetic mutation and see that particular target gene and see if anything shows up or anything doesn't. Remember, it's very sensitive. So even if one nucleotide is completely wrong, it's going to show that nucleotide is wrong, right? So we can isolate that target gene and then go ahead and sequence it. And because we've isolated it so much and amplified it so much, we can then go ahead and sequence it and see exactly what the target gene looks like if there are any mutations. We can also make it so that if the primer, right, binds to where the mutation is, right? And say we have a primer that considers a mutation, if the person has a mutation and the primer binds that it's complementary to that mutation, the primer will bind and we have PCR. So if the primer doesn't bind, that means a person, that's because the primer, remember, is complementary to the mutated sequence. That means a person does not have the mutation. So it's a very quick way to diagnose a genetic disorder, very quick way to also sequence the complete sequence of the DNA, right? If you have a genetic disorder. It can tell you the location and the nature of the mutation. It can be determined quickly. Allele-specific probes for genetic testing can be synthesized, making PCR important for diagnosing genetic disorders. It can also diagnose other kinds of things as well. So infections, right? A lot of viral infections are diagnosed with PCR, including this COVID-19, right? Basically, right now, they're sticking a nasopharyngeal swab through your nose. Basically, it's kind of an invasive procedure, honestly. It's not fun. It goes all the way up. They take some sample, and then they go ahead and run the PCR, and they run the PCR with primers that are specific to the sequence of COVID-19. So if you have COVID-19, right, the primers are going to latch on, they're going to anneal, and then they're going to allow for the extension of the COVID-19 sequence to be replicated, right, to elongate. If you don't have COVID-19, those primers are so specific that they're not going to latch on, they're not going to be able to anneal to any of your genomic DNA, because it's not COVID DNA, right? It's your human genomic DNA. So it's a very, really fast way of diagnosing these kinds of infections. It's also used for HIV, hepatitis, lots of things, even bacterial infections as well. PCR is very quick for deduction. You basically target just the sequence of the infective organism, and by targeting the sequence of the infective organism, because it's so sensitive, it's not going to target a specific. It's not going to target your own DNA. So it's not going to amplify your DNA. And if we see a band, that means voila, you have the disease, which is not good. There's also plenty of applications in DNA forensics, right? Criminology, so crime scenes. You get your DNA sample from blood, saliva, sputum, semen, everything. And you can then go ahead and compare that DNA sequence that you have, or that DNA fingerprint that you have, compare it to a database of DNA fingerprinting. The only downfall of this is that if a person committed a crime and that person's DNA was never shared, right, you have no way of detecting that person. That's why sometimes there's these things where there's traffic checkpoints and everything where a multitude of people are just randomly checked and their DNA is taken, so that this DNA database can be increased, and so we have more people to compare it to if you commit the crime. Fossils, right? So ancient DNA, DNA from single cells. So these are all, if you think about DNA forensics, fossils, DNA from single cells, all of this DNA happens in very minute amounts. It's not in a lot. It's not usually a lot of DNA. And so PCR, because it's so sensitive, can actually amplify it. So it's fantastic for that. We also have reverse transcription PCR. This uses reverse transcriptase. So we go ahead and make cDNA. So it's a methodology for studying gene expression, or essentially the genes that are expressed, the mRNA that are made, right? So mRNA production by cells or tissues, or even specific tissues that are undergoing certain conditions like cancer or something like that. So reverse transcriptase is used to generate the double-stranded cDNA, and then we do the PCR of cDNA. And then the amount of cDNA is amplified. That amount is dependent on the amount of mRNA that we had from the beginning. And so we can even figure out how much, the level of gene expression that there was in different samples. And then if you want, the cDNA that we have amplified with the PCR can be put into different libraries, into different vectors to make cDNA libraries. Then we have quantitative real-time PCR. Now, as a student, and I still make this mistake now, I get confused because real-time, to me, looks like RT, right? So don't get reverse transcription PCR confused with quantitative real-time PCR. Quantitative real-time PCR is abbreviated to qPCR, and this basically is able to tell you the amount of the original DNA that you had, right? Because even though we know that it increases exponentially, like your DNA starting from the beginning amount of target DNA that you had. After it goes to PCR, it does get cloned exponentially, get copied exponentially, but towards the last few cycles, DNA polymerase starts slipping up a little bit more and doesn't work quite as well. And so it doesn't really actually replicate exponentially as well, or as well as it did in the previous cycle. So it gets worse and worse and worse. Like it just gets tired. Think of the enzyme as getting tired. So we can't really, even though we can basically figure out a proportion of how much DNA we had at the beginning through reverse transcriptase PCR or normal PCR methods, we can't actually quantitate or give an actual number to how much DNA we had at the beginning. But with quantitative real PCR, we can. You usually include things like fluorescence with it. You basically measured how much fluorescence level there is compared to how much there was at the beginning. And then with that correlation, you can figure out how much DNA you had at the beginning after PCR. So real-time PCR allows researchers to quantify amplification reactions as they occur in real time. So as they occur, like after 10 minutes, after 20 minutes, after 30, we could see, again, this fluorescent marker. And depending on the intensity of the fluorescence, we can figure out how much DNA there is. It's quite amazing, actually. We can also, okay, so this is just, again, cDNA, right? So using reverse transcriptase polymerase chain reaction. And it just kind of underlines why it's important, because mRNA here it lacks introns, and therefore it lacks those intervening sequences that you would normally get if you were just sequencing it or analyzing or copying your target DNA directly from your DNA. Yeah. Okay. So once as well, after you get your cDNAs, you can also go ahead and run them on a gel after amplifying them through the PCR, and the complement of the mRNA of interest is identified. So you can even separate these fragments that you've made through PCR, because there's in good quantity now. You can separate them on a gel and see that, you know, that with a gel, or I believe we'll get to it. I'm not sure if we already went over it. But with a gel, you basically, you separate fragments depending on their size, and so you should know how big your fragment is, and therefore you can take it. You see, after you run it on a gel, you can see where your fragment stopped. You can say, okay, that's my fragment. That's 1,500 base pairs long. That's my fragment. You literally cut it out of your gel and then isolate it. So you take away all the gel components, the agarose, polyacrylamide, whatever, and isolate just the DNA. So PCR is incredible, like the uses that we have. But again, don't forget that sometimes there are limitations to the PCR technique as well, just because it's overly sensitive. We do need a little bit of information, right? And it is limited by the size of the DNA cloning. So a lot of times we can substitute molecular techniques with PCR, but sometimes still those cloning vectors are important, especially when our DNA fragment is too big. So that's where we're at today, and we'll pick up again on molecular techniques for analyzing DNA later. Okay.